Hey guys, so today we are focusing on meiosis. All right, our objective and standard are still the same. If you'd like to write those down, go ahead. All right, so our essential questions for today is what is responsible for the diversity of life and how is genetic information carried from one organism to its offspring? You should be able to answer these two questions by the time we get to the end of our video notes for part 2 tomorrow. So, starting off with chromosomes. Okay, homologous chromosomes are the two corresponding chromosomes that carry the same genes. Okay, so one set of chromosome comes from each parent. Okay, they're similar but not identical, but they carry the same genes in the same order. Okay, and alleles for each trait may not be the same. All right, and alleles are like two different options for a trait. For example, if we're talking about the trait of hair color, okay, two different options, maybe brown hair, that would be one allele, and blonde hair would be another allele. So meiosis is the process by where a single cell divides twice to produce 4 daughter cells that contain half the amount of chromosomes of the parent cell. Okay, so meiosis creates 4 sex cells called gametes. Okay, a sex cell is just gamete is just the fancy name for a sex cell. Okay, male gametes are sperm. Okay, female gametes are eggs. All right, diploid cells is what we have to concentrate on first. They're non-reproductive body cells that are called somatic cells. We have gone over somatic cells before. All right, somatic cells are diploid because they contain 2 sets of chromosomes. This is represented by 2n, which would be 2 multiplied by the number of chromosomes. 1 set comes from each parent, and human diploid cells contain 46 chromosomes. Haploid cells are what we refer to as having half the number of chromosomes as a body cell. Okay, so they're represented by n. Okay, that would mean half of the number of a normal cell. Okay, so human gametes each contain 23, which is half of 46. Okay, so N would be 23 for humans. All right, in fertilization, 2 haploid gametes fuse together during fertilization to create a diploid cell. Okay, so 23 comes from one parent, 23 comes from the other parent, they come together, they create 46. Okay. So in the picture, you can see that the sperm cell has 23, the egg cell has 23, and then 2n would be 46 because they come together. Okay, when a cell is fertilized, it becomes diploid and it's now called a zygote. So we're going to calculate the number of chromosomes. So go ahead and calculate either the n value or the 2n value. For example, for a human, n would be 23. And then 2n, so 2 times n, okay, would be 46. So 23 times 2 would give us 46. So go ahead and calculate the diploid number for amoeba and the pigeon, and then the haploid number for a chimpanzee and onion, okay? If you're looking to go from diploid to haploid, all you would have to do is divide by 2 instead of multiply. So, reduction division. Meiosis is called a reduction division process. Okay, in humans, the starting cell has 46 chromosomes and the ending cell has 23 chromosomes. Okay, this just means that the chromosomes are reduced by division. Okay, so the starting cell has 2n, which equals 46. Okay, and the ending cell has 23, which is half of 46. Okay, so 46 divided by 2. Okay. Meiosis in general, meiosis creates a genetically unique gamete with half the number of chromosomes as the parent. Okay, sexual reproduction makes sex cells, aka gametes. Okay, it makes haploid cells. Okay, they divide twice, so one divides into 2, and then those 2 each divide into 2 again. Okay, to create 4 haploid cells from 1 diploid cell. Okay, this happens in meiosis 1 and meiosis 2. Okay, in interphase, we've already covered interphase, okay? Just like in mitosis, interphase has to occur before meiosis takes place. Okay, during the S phase of interphase, DNA is replicated just like in mitosis. Okay, Each duplicated chromosome consists of 2 identical sister chromatids attached at the centromere. You can see that in this picture here. Next to it, all right, the centromere is the little teal part in the middle, okay? And then we have each chromatid on the side, all right? Centrioles also replicate, okay? Which is the thing that the spindle fibers come out of. All right. So in our overview of meiosis, okay, there are two main phases of meiosis, meiosis 1 and meiosis 2. Okay, in meiosis 1, one cell divides into 2, and then in meiosis 2, 2 cells each divide into 2, creating 4. All right, so cells divide twice in meiosis, and interphase occurs before meiosis 1. All right, meiosis takes place in 2 stages of division, meiosis 1 and meiosis 2, which means PMAT, aka just like in mitosis, prophase, metaphase, anaphase, and telophase occur twice. Okay, once in meiosis 1 and then once in meiosis 2. All right, we are going to get through prophase 1 today, and then we're going to cover the rest of it tomorrow. Okay, so in prophase 1, each replicated chromosome pairs up with its homologous chromosome. Okay, we've covered homologous chromosomes already. Okay, they are like matching chromosomes basically. Okay, this forms a tetrad, okay, containing 4 chromatids. We know that half of a chromosome is a chromatid. Okay, spindle fibers form, and it's the longest stage of meiosis. It is 90% of meiosis. Okay, so a tetrad is, as you can see here, let me get my pen. Okay, you can see here that a tetrad is 2 chromosomes together, okay, and they're a homologous pair, and then a chromatid would be half of these. So we have 4 of those chromatids. All right, homologous chromosomes complete the crossing over in prophase 1. This is like the most important thing that happens in meiosis, okay? Crossing over is the physical exchange of genetic information. Okay, let me move this. Okay, synapsis holds the chromosomes together at a location called the chiasma. Okay, so just right here, okay, where they're crossing over together, okay, that's the chiasma, that's what holds them together. All right, so they're held together by the chiasma. All right. In prophase 1, crossing over produces new combinations of genes, which accounts for genetic diversity, okay? It's why we don't look the exact same as our sister or brother, okay? Even though we have the same 2 parents. We may have the same 2 parents, all right? In prophase 1, we have this process. We start with a homologous pair, okay, with the synapse connecting it. Okay, and then after crossing over, you can see that a blue section is on the red chromosome and a red section is now on the blue chromosome. Okay, they literally just exchange pieces of each other. Okay, which is that mixes up the genes. Okay, and that is why we don't look the exact same as a person who may have our same 2 parents. Okay, that is all we are going to cover for today. All right, tune in tomorrow for the rest of meiosis. All right, have a good day. See y'all later.